is Stefano Gaiarsa. I'm from the San Matteo Hospital in Pavia, Italy, which is a city next to Milan. And my work is about a way to measure genome plasticity in bacteria. So we keep hearing about bacteria that keep acquiring novel genes, especially for antimicrobial resistance. And we hear that they do it at a very fast rate. And so our question was, how fast are they when acquiring such genes? Can we actually measure such speed? And also, are there groups of bacteria also within the same species that are able to acquire novel genes of whatever with whatever function at different rates? So we called this rate, this speed, we call it plasticity, we call it genome plasticity, and we looked in the literature for indices that we could use to measure that, but we actually found only indices that were based on static genome content, like indices that could be used to measure pangenome size or something like that. So we had to come up with ours. And actually, we come up with one which is called FOGS, flux of gene segments. This is the formula, but I'm going to explain it very quickly. So FOGS is an average, a mean, actually, among pairwise distances of genomes of the same group. And these pairwise distances are measured as gene content distance over evolutionary distance. Gene content distance is measured, it's calculated using graphs, and this method also takes into account that genes can move together. So we don't just count genes. We consider the fact that a couple of genes that are in the same genomic region can be acquired and lost together in one single event, while evolutionary distance is measured using core SNPs. We put our novel index to the test with a selection of high-quality published genomes. I'm going to talk briefly just about Klebsiella pneumonia. We took our data set and we divided into clusters using FastBabs. And for sake of simplicity, I will refer to clusters using the corresponding multi-locus sequence types, because in Klebsiella they're almost the same thing as FastBab clusters. And what we actually discovered is that, yes, there are groups of bacteria, there are clusters that are way more rapid, way more plastic than others. And actually, those clones, let's say, to use a clinical term, that are widespread, they are the most successful worldwide, are those with the highest FOGS levels. Actually, the one with the top level of FOGS is ST307, which is the most common worldwide, which has, this cluster has more than twice the FOGS value of ST258, which used to be the dominant one until some years ago. So our hypothesis is that a higher capability of exchanging genomes quickly is among the reason of the success of this clone. And this is, the same can be applied also for the other species that are reported here in the poster. We have Staph aureus and E. coli. And on top of this hypothesis, we suppose also that we could use this index the other way around. We could, using this index, we could predict clones that are going to be successful. So maybe this index could also be used as a surveillance tool, maybe, at some point. As a side note, we studied the relationship between FOGS and antimicrobial resistance levels and virulence levels. And we discovered, at least in Clevnumo, that FOGS has a positive correlation with antimicrobial resistance levels, which we could not find for what concerns virulence. We tried to explain this by considering that antimicrobial resistant genes often also carry a high metabolic load for cells, and that the pressure of the presence of antimicrobials is not constant. I mean, bacteria are not always immersed in antibiotics. So the best way, the most successful way to deal with antibiotics would be to keep losing resistant genes and acquiring them back. So in the end, those clones that are more able to keep exchanging materials and to lose and acquire resistant genes back are those that end up being multi-resistant. So there's a couple information more on the poster, and I invite you to look up the online version of the poster. So if you have further questions, you can look up the online version of the poster, which contains some other information, or you can also look for the paper, which was published in Microbial Genomics in the meantime. The first author is Greta Bellinzona, which I really want to thank because she did almost the entire set of analysis. And also, if you have any more questions, please, you can find the contact on the poster or write us on whatever means you want.